Alright, continuing with the introductory videos. This one is about as introductory as it gets. By now, you should have a pretty good handle on what exactly the scientific method is, but I thought I would go through it really quick. Before I get started, I should mention that there are as many ways to phrase this as there are species under the sun. So, if I don't use the exact words you're familiar with, don't be alarmed. There are a bunch of different ways to phrase it. In fact, most textbooks now are moving towards the scientific process as just that, a process as opposed to a step 1, step 2, step 3 kind of thing, which we will go into. But I just wanted to give you some terms and the basic idea of exactly how this goes. So, scientific method, while it sounds like a big scary term, complicated thing, whatever, it's really just how you research a problem. So anytime there's an issue or something you don't understand, this is the process that you go through in order to figure out the answer. It can be something as simple as why does a watched pot boil slower than an unwatched pot? Or it can be something as complicated as the effect of a catalyst on the rate of a particular reaction. So it spans the full gamut. Any scientific problem, if you will, always begins with an observation, something that you see that spawns a question of some kind, something that you want to know the answer to. The classic example is the leaf falling. You observe the leaf, it falls. That usually, again, spawns some kind of question. Why does the leaf, it fall? So your second step, quote unquote, in scientific method is usually to generate some form of a hypothesis. I think the leaf, it falls because some force is pulling it down towards the ground. In the beginning of your scientific education career, the hypothesis is usually defined as an if this, then that kind of statement. But as you get deeper into your science classes, you will discover that that particular formula does not always work. To shoehorn a hypothesis into that format is unnecessary. But it is a good place to start when you are just beginning to formulate hypotheses. The goal of a hypothesis is not to be correct, actually, contrary to popular opinion. A good scientist always tries to prove themselves wrong. So if you get all the way through your scientific method process and you realize that your hypothesis was completely and totally bogus, that's fine. Some experiments may not have a hypothesis per se, but more of a question that needs to be answered. But typically, before you begin any kind of experiment, you want to make a prediction. What do you think is going to happen throughout this endeavor? So once you've got your hypothesis, you've seen something that has sparked your curiosity, you have made a prediction or asked a question, come up with a prediction about what you think will happen as you begin to probe this particular strange occurrence. What next comes is to design an experiment. How are you going to test this idea? This can range from an extremely complicated procedure that has 35 steps. It could be something extremely simple. But the big thing about designing an experiment is that it must be controllable, for lack of a better word. It needs to be specific enough that you can control as many bits and pieces as possible. Within your experiment, you usually have variables, things that change, 2 of them in particular. The independent variable and the dependent variable. Bah. I never can spell those words. See, autocorrect is ruining my ability to spell. So your independent variable, think as this, it is the independent thing. It's the thing that you change with your own 2 hands. Your dependent variable depends on the independent variable. So the classic bean sprout experiment, when you're trying to grow a plant and see how the height varies with amount of sunlight. So the thing that you're changing, the independent variable, would be the amount of sunlight. Notice the one thing that you are changing, because with the independent variable there can be only one, no more. one thing that you are changing, in this case the sunlight, and then your dependent variable depends on that independent variable, so the height of your bean sprout. This is usually the thing that you are measuring, the thing that you are looking for. And that's another big part when you are designing an experiment, is with that data, you have to have it. You have to have it, the data. If you cannot gather data, it is not a well enough designed an experiment. This data can be what is called qualitative. So think qualitative quality. These are things like color, touch, taste, smell. No numbers involved. More sentences than it is what you would typically think of data. Ideally, we want to have quantitative data. Think quantity, numbers. So you must be able to gather data in some form or fashion. Oh, I forgot something else with your experimental design. Independent variable, dependent variable, I forgot to mention controls and constants. The word control can have two different meanings, kind of depending on the situation. A control usually refers to your baseline. So, for example, back to the plant analogy. Plants on average get, 7 hours of daylight if you've got them square in the middle of the sun. So that would be your control, your baseline. And you would compare all of your results back to this control. That's the definition you should probably stick to, is your control as a baseline to which all other results are compared. Gives you an idea of like the expected. Constants, on the other hand, are things that do not change. Things you hold, you guessed it, constant. So with the plant example, this would be the amount of water you give them, whether or not you give them fertilizer, the amount of dirt in the pot, maybe even the size of the pot. All those other little pieces that you hold consistent throughout the experiment, so that there is but one thing that you are changing, but one dependent variable that you are looking for. Now, sometimes, as you begin designing this experiment, something doesn't work. Maybe you're not getting the data you expected. Maybe you're not getting any data at all. So it requires you to go back and redesign your experiment, perhaps. Maybe you need to add more water. Maybe your plant isn't growing at all. Maybe you do need to give it a little fertilizer, a little kickstart. Maybe as you start working on this experiment, you realize that your hypothesis was completely and totally off base. So perhaps you have to go all the way back up here. Reformulate that hypothesis. All of this is fine. Encouraged, in fact. Because science is all about that that searching for greater understanding, kind of whatever forms it takes. And oftentimes, it's not the form that you expected. So perhaps you go back, reformulate your hypothesis, tweak your experiment a bit, gather some more data. Now we're making some progress. It makes some sense. After we've got some data, we need to analyze it. We need to go through, talk about are there averages, trends, graphs, things like that are all good things to have in any kind of scientific report. But all in all, you're drawing conclusions. Was your hypothesis supported? Yes or no? Remember, any good scientist is trying to prove themselves wrong. To be right earns you no prizes. In fact, you may find yourself kind of forcing the data to fit the conclusion. Don't do that. In fact, like I said, if you're wrong, it gives you more to talk about, more things to look at. And so that's why I added this last step of evaluate to the bottom of this scientific process, is you need to stop and think about your experiment. How good was it? Were there things you should change next time? Maybe something else you should have done? Was there bias present in your experiment? Check yourself. Were you trying to force data to fit conclusions? And so all of this process is more of a cyclical than a straight up and down line like I have drawn here, but it can be a lot of fun. We started doing a science fair type project here in my classroom, and all of a sudden we get 3/4 of the way through the project and nobody's experiments were working. All of them seemed to be going awry. And they kept saying, but I have no data. Nope, that is data. Even a failure is a result. It is something, a conclusion that can be drawn. So please don't be afraid of your experiments not going as you planned. In fact, those are usually the more interesting ones. So I encourage you to explore, do experiments, really spend some time thinking about that lab that you're doing in class. Don't let it just be a step 1, step 2, step 3. Think about it. The world is a pretty cool place, and fact is often stranger than fiction. So happy hunting to you.